Oh, sorry, is that still on? It's still on. So, good afternoon. I'm going to probably come at you from a whole different level than you just heard with Don's presentation, talking and focusing really more on how to synthesize all the information that you've been getting into a framework that's going to lead to positive steps in terms of policy and management decisions. My name is Tom Grovhoug. I'm with Larry Walker Associates. I'm here today on behalf of Sacramento Regional County Sand District, also known as Regional Sand, and other municipalities who treat wastewater and discharge treated effluent into the Delta and its tributaries. Dr. Tim Mussen, who's a staff scientist for Regional Sand, will be providing additional testimony after my initial remarks. I really have three key points that I would like to touch on today. The first is to describe the mission and role of the public entities that treat wastewater in the Delta. The locations of the publicly owned treatment works are shown on the map that is now being displayed. There are, I believe, 17 publicly owned treatment works that discharge either in the Delta or in the immediate vicinity of the Delta. Total permitted discharge from those facilities is around 330 MGD. Sac Regional is the largest of those facilities. It's currently permitted at 181 MGD. Sacramento is a secondary treatment system. Most of the rest of the POTWs that are displayed on the map are already at a advanced secondary or tertiary level. And when I say advanced secondary, I mean secondary with nitrification, most of them with denitrification, which would take total ammonia from probably 30 total nitrogen, total nitrogen from 30 to less than 10, probably in the 8 to 10 range. So that's kind of the picture of current wastewater treatment. Really drawing from the Regional Sand's mission statement as an example, I think it's a good way to understand the mission for the folks that operate these facilities, and that their common mission is to protect public health and the environment through reliable and safe conveyance, treatment, and disposal of wastewater in the most cost-effective manner possible now and into the future. The people who manage and operate these municipal facilities take their role in environmental protection very seriously. They strive for and often achieve 100% compliance with the tough discharge requirements that are established by the Central Valley Regional Water Board. They also participate heavily in scientific and policy efforts in the Delta. To name a few, the Delta Regional Monitoring Program, the Delta Drinking Water Policy, the Delta Methylmercury TMDL, also, as was presented at the last flow workshop, the San Francisco Bay Nutrient Management Strategy, CV salts, and a number of other collaborative environmental initiatives. Their role, working with other stakeholders, is to understand the impacts of their discharges and to take action as appropriate. Their job is to ensure that the public resource expenditures that are made for wastewater treatment will create a benefit in the Delta environment and to the protection of beneficial uses. So as such, wastewater representatives are a valuable asset in the Delta science, policy, and management arena, and most particularly with regard to topics such as water quality, the Delta food web, and toxicity. This leads to my second point, which is the wastewater community is very supportive of the efforts that are embodied in this workshop today and others like it that are aimed at the restoration of the health of fish communities and the Delta ecosystem in general. Clearly, the severe declines in populations of multiple fish species serve as a call to action to all parties, both within the Delta and those served by the Delta. There is a clear need to come together to take an integrated, system-wide approach to solve these significant problems, and understanding the role of delta flows, residence times, hydrodynamics, in combination with other factors, is an obvious and necessary piece of that analysis. So my third and probably main point is that complex problems of this magnitude call for the combined best efforts of all key players around the table, that is, scientists, policymakers, and managers, working together with a common set of goals and objectives. It's important that these efforts be organized in a framework that promotes effective collaboration, participation, and decision-making. And unfortunately, that hasn't always been the case in situations in the Delta. At the last Delta Flow workshop, Lauren Hastings of the Delta Science Program provided an excellent presentation which outlined a concept that she called collaborative adaptive management. We are aware that that approach is currently being applied to help resolve some of the issues related to the biological opinions. The keys to that approach include the credibility and relevance of science, legitimacy of the process, and a focus on outcomes. And another key is to get the right people involved at the outset. The wastewater community is very supportive of these concepts, sees many similarities with some of the other successful collaboratives that they have been involved in California, some of the ones I named previously. We strongly encourage this panel, if you haven't already, to view the video of Ms. Hastings' presentation that's available from the Delta Science Program, to confer with Delta Science Program staff, and to consider such an approach as kind of an overarching framework for your recommendations that you'll be making. We believe there's really no time to waste in moving forward with such an approach, and stand willing and ready to participate in such a process, confident that it will lead to better solutions. So with that, as an overview, I want to hand it over to Dr. Tim Mussen.
Thank you, Tom. So as a quick outline for the remainder of this presentation, I'm going to put forward some preliminary results from a collaborative USGS experiment that Regional SAN has been participating in, looking at growth of phytoplankton in the Sacramento River in the presence and absence of wastewater effluent, and also another study that we looked at for pyrethroid pesticide removal within the wastewater treatment process. And then I'll go on to discuss a few factors that are limiting phytoplankton abundance within the system, and discuss a few potential river management practices that could support native fish survival and food abundance. So last fall, USGS conducted a Lagrangian-style experiment that started up at the I-80 Bridge in the northern part of Sacramento and traveled with a water parcel down the river past the Sacramento Regional Wastewater Treatment Plant and continued further down until Isleton, which is about the area where it connects up with the Delta. And the goal of the study was to look at phytoplankton growth within the presence and absence of river water or wastewater effluent. And so the way that we created these parcels of water with no wastewater present was by diverting the wastewater into storage basins for 17 hours at the treatment plant, creating roughly a 6-mile stretch of river conditions that was wastewater-free. And then one boat would remain within that parcel, and another boat was stationed 24 hours ahead of the parcel in an area which had normal discharge conditions, and both of them tracked these parcels for 5 days down the stretch of the river, collecting samples each day to look at how the phytoplankton and the nutrient concentrations were changing over time as a comparison. So to give you an idea of the results here, this first graph is for ammonia, and this is the travel time of the boat with 0 marked as the location of the discharge. So here, upstream of the discharge, we see that no ammonia was present in either one of the parcels, which is the usual condition. And then orange down here represents the wastewater-free parcel, and we can see that the diversion was effective, and the ammonia didn't slosh back into that large parcel of water throughout the experiment, but ammonia rates stayed low. And then when we look at the regular mixed effluent, we see that ammonia concentrations were much higher and remained, maybe there was some decrease throughout the experiment, but was still present all the way until the location where it met up with the delta. So when we want to look at how the chlorophyll was responding in the presence and absence of wastewater, in general, there was no significant difference between these two parcels. However, we did see that the Chlorophyll-a concentration did show a decline in both parcels as it was moving down the river. And this leads us to believe that there's some other factor that's likely causing this phytoplankton reduction besides wastewater effluent. So during the experiment, Nicole Travis, who was working with Alex Parker, was collecting samples of river water and putting them into Cubitainers to create mesocosms. And they were drifted next to the side of the boat. And I would like to mention that Tamara Kraus was the PI for this investigation from USGS, and that it was funded by IEP as well as Regional SAN. Get that in. So in one Cubitainer, it was collected from upstream and has no effluent, and another Cubitainer was collected just after the wastewater was discharged, and a third trial was conducted where some ammonia, not the whole effluent, but just ammonia, was added in to see what its effect would be on the phytoplankton. And here, when we look at how these changed, they were sampled at the same frequency as the river, drifting downstream, and there was no ammonia within the upstream non-effluent sample. And then here's the 40 micromoles per liter that was declining over the 5 days. And here's the wastewater, which at the time had quite a high concentration because we're in drought conditions and have been since the fall, so the river flow was pretty low. And we see a bit of a decline here as well. So the nitrate within all three of these Cubitainers started off at relatively similar levels. But then halfway through the experiment at 50 hours, we can see that in the mesocosm without added nutrients, the nitrate rates have fallen down to pretty much non-existent. Whereas in the mixed effluent or in the added ammonia, the nitrate levels remain relatively high throughout the experiment. So when we look at how the chlorophyll responded within these floating mesocosms, we actually don't see too much of a change, maybe a little bit of a change here in the upstream river conditions that then stay constant throughout. But in both of the mesocosms where nutrients were added, we see this continual increase in Chlorophyll-a concentrations throughout the entirety of the experiment, up to levels which are sometimes considered blooms when we see them down in Sassoon Bay. And the difference between the river, where the samples were being collected, and then the mesocosms were a couple-fold. So one is that the mesocosms were floating up at the surface, so they had more light than the phytoplankton that were being dropped down into the bottom of these channels. And the other difference is there were no clams in the mesocosms, whereas there probably was some grazing occurring throughout the river system as the clams were cycling down near the bottom of the river. We also were interested in what species were growing in the river in the presence and absence of wastewater. And so we collected enumeration samples. And we wanted basically the desirable phytoplankton that Brian was pointing out earlier, generally considered to be diatoms. So these are charts showing what the diatoms, the percentage of the samples of diatoms were like. And in terms of biovolume, the diatoms were generally above 80%. So most of the biovolume in each one of our samples was comprised of diatoms. But diatoms are a lot bigger than most of the other phytoplankton out there. So if we're just looking at cell counts, we can see that this biovolume for the cell counts for diatoms was a fair bit lower, maybe around 30%. And the other thing to notice is that in the wastewater present and absent, there wasn't really a change in the ratio of diatoms to other phytoplankton species. They look similar regardless of how you enumerate. And when it comes to the amount of food that's available for fish, it seems as though biovolume would be a better indication as the total quantity that's available for them to eat, rather than just the total number of particular cells that happen to be in the environment. So now I'm going to move on to another study that was conducted at the Sanitation District. And as we just had Dr. Weston present pyrethroid issues, I thought you might be interested to know that pyrethroids do exist within sewage coming into the wastewater treatment plant. And what we would recommend for removing them from the aquatic environment is greater source control, such that less are being put into the environment. But it turns out that wastewater treatment processes also do decrease pyrethroids, and generally by 90%. So the dark bars are the influent, and that's basically the untreated sewage coming into the system. And then when we look at our current secondary level of treatment, there's been roughly a 91% reduction here when we're averaging up amongst the 6 different pyrethroids that were tested. And then at the time, we also had running a pilot experiment for the biological nutrient process that's going to be used into the future by the wastewater treatment plant to remove nitrogen from the system. And under the enhanced treatment, we saw even a further reduction of pyrethroids. So, now to discuss some of the factors that are likely to be limiting phytoplankton within the delta, a new paper from Kimmerer and Thompson came out this year that actually looked at the combined grazing rates of clams and microzooplankton. And what it showed in the low salinity zone is that during the spring and summer and in small and large channels, the amount of phytoplankton produced, that's shown with the bars with the P, is lower than the amount of phytoplankton that's being consumed, which are the bars with the C's here. And that was in every time and location that were tested. Now in the consumption rate, the bottom is also respiration. So when the phytoplankton are down out of the photic zone, they're going to be burning their own energy. And then they're also getting lost to consumption. But that in total was showing that phytoplankton experience a net loss in the low salinity zone, and they require subsidies from elsewhere to keep their biomass at the low levels they're at to try to sustain pelagic life. And it turns out that clams aren't just in the low salinity zone. People know clams are distributed throughout the delta. And this is a paper from Lucas and Thompson showing how clam grazing rates are being calculated based on their biomass and also the water depth throughout the system. So when you get into their take-home message is that when you get into shallow water depths, you do have potential to grow a lot of phytoplankton when there's no clams present, but if clams are there, they become areas of net loss. So you really need to factor in both the abundance of clams as well as their distribution. Now you see going up the Sacramento River there's no bars, and that's because people haven't really looked there for clams. So we decided to go see if there were clams in the Sacramento River, and here's an example of a 1-minute trawl on the bottom pulling up Corbicula, and yes, there's clams in the Sacramento River. This is still early data. We're trying to find all the biomass from them. It takes a while to measure. So one area that we could increase productivity by having that shallow water depth and long residence time is actually within floodplains. Because as the water becomes inundated, the phytoplankton will grow and the zooplankton will grow, but the clams don't have time to colonize. And therefore, the energy can be transferred into food for fish. And native fish happen to be better at getting onto these floodplains and off again because they evolved in this system that had a variable climate compared to most of our invasive fishes. And the other benefit that comes from floodplains is that they promote a diverse life history strategy for the salmon moving in, so that not only are they getting some food and growing faster, but then they're also leaving at different times to head out to the ocean, and perhaps it'll line up better with when the oceanic conditions are optimal for the Chinook to arrive. The Yolo bypass is a particularly important floodplain because it drains down into Liberty Island and then into the northern delta, and that is the area where we still have the highest amount of native fish living within the delta system compared to, say, the central or the southern delta. But one thing that's important to remember when you're considering floodplain restoration is that there does need to be some refuge against avian predation. So some deeper pits, or at least some structure, would likely be helpful. And then I also wanted to discuss the management option of using guidance devices to try to keep fish within the right course. There's been large agreement over this workshop that fish heading into the central delta don't do well. And actually, John Barrow briefly discussed this during the end of his presentation before, where there's currently a floating fish screen that's a barrier placed at the front of Georgiana Slough, so that the fish that are following that shoreline will hopefully be deflected out into the middle of the river and stay in the Sacramento to provide greater survival rates. So the main messages from my presentation was that Sacramento River phytoplankton growth is similar in the presence and absence of wastewater effluent, and that effluent can actually increase phytoplankton growth when they're placed in Cubitainers with higher light availability and less clam grazing. Diatoms do dominate the phytoplankton biovolume, but when you look at them in terms of cell counts, they look less abundant. Source control is the preferred method for pyrethroid removal, but wastewater treatment is also effective at removing these pesticides. And in the future, as we go to our biological nutrient process, we'd expect the reduction to be increased. Light limitation and grazing losses can limit phytoplankton abundance in the low salinity zone and likely throughout the rest of the delta, and floodplain inundation and behavioral guidance devices may offer some good management techniques to promote fish food abundance and survival. And so I'd also like to thank the members of the Lagrangian Transect study and the Clam Abundance Survey crew. Thank you.
Do you have time for a question or two?
I've got a quick question. In the cases where wastewater treatment could remove pyrethroids, where does it go?
So it does get sequestered into the sludge that is grown and then into the digesters. And then it varies by which treatment process as to how people use or deposit the collected sludge.
But it's not totally chemically inactivated or anything. I mean, it could still function if the sludge is used inappropriately or something.
Yeah, well, we haven't. It does not go into the aquatic environment, right? So ultimately, I believe that I don't know how the pyrethroid's chemical nature is going to change within a digester or then sitting within a treatment pond for 5 years. And then at that point, it might end up being landfilled. There's multiple pathways that ultimately dispose of the process.
Thanks.
I have a question about the primary production stuff. I assume that the experiments were somehow water was screened to remove microzooplankton before.
Before the Cubitainers?
Yeah.
Yeah. I'm not entirely sure if Nicole had filtered out the larger organisms before putting them into
that's the first question. The second question is, as I recall, Dick and Francis's work was mostly in Sassoon Bay,
right,
with with diatoms from 2 and up salinities. Do you think there's any likely to be physiological differences for freshwater diatoms compared to estuarine diatoms?
In terms of nutrient
response to nutrient.
Yeah, so from what I believe, nutrients, most phytoplankton sequester ammonia before they get into nitrate. And that's going to be for a marine or a freshwater diatom. So when ammonia is present, it'll end up being collected first.
Okay. time for a break. Let's come back at about 10 after.